In the previous videos, we ran a full analysis and exported the results. In this final video, we'll cover what to do when the results need refinement. There are two common situations where you'd want to refine your results. The first, when a specific cell mask is incorrect. For example, two adjacent cells might have been merged into one, or one cell might have been split into two. In this case, you can directly edit the mask to fix it. The second, when the lipid droplet detection is too strict or too loose across the whole dataset. For example, the refractive index threshold might have been set too high, causing many real droplets to be missed. In this case, adjusting the parameter and rerunning is the better approach. Both cases start from the same place, opening a saved history for editing. From the result history list, select the history you want to refine. Click Review Analysis or History Run to open it. This is the same interface as Test Run, but now you're working on stored results rather than running from scratch. Any changes you make can be applied and saved as a new history, leaving the original untouched. Let's start with the first case, fixing a cell mask. Suppose two adjacent cells have been merged into a single label when they should be separate. You could try to redraw the final cell label directly, but there's an easier way. In this pipeline, the final cell labels come from a chain of processes. The cells are first identified as seeds, small regions marking each cell center, and those seeds are then expanded outward to form the full cell labels. This means if you edit the seed mask instead of the final label, all the downstream processing happens automatically. The pipeline re-expands your corrected seeds into properly separated cells without manual drawing of cell boundaries. In the Output Navigator, find the seed mask used by the pipeline. Open the mask editor by selecting A-8, Cell Seed Labeling, in the Output Navigator and clicking the Editor button. The mask editor offers several tools. The most basic ones are Brush and Eraser. Brush adds to the mask, and Eraser removes from it. Beyond drawing, there are also tools for working with labels. Merge combines two separate labels into one, and Divide splits a single label into two. For 3D editing, there's the Interpolate tool. Draw a mask on one lower Z-slice. Then move to another Z-slice further above, and draw an additional mask. Once the masks are defined on both slices, click Interpolate. The mask editor automatically fills in the slices between them, building a smooth 3D shape that connects your two drawings. For our case, where two cells were merged into one seed, we'll delete the seed mask first. Select the Merge Seed label. Then click the Delete Label tool to remove the incorrect label. Next, use the Brush tool to manually draw separate seed labels for each cell. You can scroll through Z-slices while editing to make sure the separation is consistent in 3D. When modifying a specific label, make sure to click the corresponding label mask to select it. When you're done editing, click Apply Changes to save the edits to the mask. Now we need to push these changes through the rest of the pipeline. Click Transfer All to apply the edited mask to all downstream processes. Then click Execute All to regenerate the affected outputs. Once it finishes, return to the live view. The pipeline has now expanded your two separated seeds into two properly labeled cells, and all downstream LD measurements are updated to reflect the corrected cell labels. Verify the result in the live view. The two cells should now be correctly separated, each with its own color label. The cell mask is fixed. Now let's look at the second refinement case. Suppose the lipid droplet detection looks too strict. Visually, you can see many bright droplets in the HT image that the pipeline failed to label. This usually means the refractive index threshold was set too high. The simple fix is to lower the threshold and rerun. click the E-1.LD segmentation_RI process to see its parameters. The L threshold value is the lower refractive index cutoff. Let's say the current value is 1.37, relatively strict. Lower it to 1.365, the default. Now click Execute. And then click Execute All to rerun the whole pipeline with the new threshold mask. When the pipeline finishes, check the live view. You should see more lipid droplets detected than before, including the ones that were previously missed. The parameters are now refined. Let's save this refined version as a new history. Click Save History in the toolbar. A dialog opens asking for a name. Use a descriptive name that captures what makes this version different. For example, RI threshold 1.365 with corrected cell mask. A clear name makes it easy to find this version later. Select the outputs you want to save. You'll also see an option called Set as Main History. When this is enabled, the new history becomes the default version displayed in the result history list and used for export. Enable this if the refined version is the one you want to treat as your primary result. Click Save. The new history now appears in the result history list, alongside the original. Both are preserved. You can always compare them or go back to the original if needed. From here, you can export the refined results the same way you would export any other history. Right-click the history, select Export Histories, and follow the export process from the previous video. That covers the main refinement workflows in TomoAnalysis. Whether you're correcting a specific cell mask or adjusting parameters across the dataset, the pattern is the same. Edit, regenerate, save as a new history. This concludes the TomoAnalysis tutorial series. Thank you for watching, and happy analyzing.